Right, I'm on, so admit a new student. So as I call up this first whiteboard, if you are a note taker, which is not a bad thing if you are, I'm gonna try to start where I left off from last time, which is membrane potential. So I'm just gonna write that term down. This refers to what happens along the edge of a plasma membrane of a cell. And in this case, we're talking about a neuron, a nerve cell. And when we separate charge across a membrane, meaning one side being opposite from the other in terms of charge, that creates the potential to do work or the potential for energy. And we're talking about the plasma membrane. So that's what its literal definition is. Now, in practical purposes, how this happens, how we set up this membrane potential, is that there are three types of membrane channels that may contribute to this situation. I'm going to decipher this in real speak for you. Membrane channels, just think of proteins. There's proteins on the membrane. So if this is a cell membrane, this is the inside and this is the outside of that membrane. A channel is just a protein, basically, that sits on this membrane, and it allows things to go in and out. What activates this channel, this gate opening and closing, is what defines these three types. And the three types are chemically gated, voltage gated, and mechanical gated. So there's chemically gated channels, voltage gated, and mechanically gated. And the nervous system uses all three of these types of channels, these proteins on the surface. All they refer to is what activates their gates opening. In this one, it would be chemicals. Sometimes they're called ligands, L-I-G-A-N-D. Just ligand just means a chemical signal. So, or a signaling chemical is a better way to put it. So chemically gated would be if a chemical, I'm going to call it a different color, if a chemical binds to this protein, it opens a gate. Voltage gated, what that refers to, if there is something that changes the voltage around this membrane, it opens the gate. And mechanically gated, think of the most obvious analogy or example I can think of is the sense of touch. Some nerve endings are activated, AKA their protein channels open, when they are mechanically or physically depressed. There's some sort of a mechanical trigger on this that also opens a gate. So that's what those three types refer to as their activation. Now, when a nerve cell, the next topic is called resting membrane potential. And I believe that I went over this very quickly before we stopped last time. Here is a motor neuron nerve cell. This is a typical motor neuron. We went over the anatomy of it in our discussion last time. These are dendrites near the soma or cell body. There's a nucleus in here, which is it's not that important that I draw it. I just draw it to remind you that that's the cell body. These dendrites receive impulses. This is the axon that generates and transmits an electrical impulse. When we talk about this cell at rest, we use a term called resting membrane potential. That just refers to what's the situation of this cell's membrane when it's at rest. And the situation is this. Due to several factors that unfortunately I don't have time to go into all of them in detail right now, but due to several factors, there is a high concentration of sodium ions outside of the cell and a high concentration of potassium ions inside of the cell. Now that just occurs right at the edge of the membrane, not deep within the cell here and deep outside of the cell here, but just along this border we get a separation of these ions. There are different gates or channels that help create this situation. And then there is a pump on the surface of the membrane of all of our cells called a sodium-potassium pump, and it uses energy, it's ATP driven, that maintains that separation of ions. All right, this next part might be a little bit a little bit weird to grasp if it's the first time you've heard this story, but sodium happens to be a much stronger positive ion than potassium. So it's stronger in its positive charge. Because of that, we say that during rest, there's a high concentration of sodium ions outside, a high concentration of potassium ions inside, and the net charge that's created by that is positive to negative. The reason that is, it's the relationship of the outside compared to the inside. The outside is more positive than the inside is. So when we stick a probe in here inside of a nerve cell, it reads negative 70 millivolts. Don't worry about millivolts and all that electricity business. Just know that the inside is negative, the outside is positive. That's a net charge, and it's due to high concentration of sodium out, potassium in. And that's all the way around this membrane, all the way around the cell, the whole cell. So that's when the cell is at rest. I almost said doing nothing, but it's not doing nothing when it's at rest. It's actually creating neurotransmitter and storing it down here to be released. But when the cell's at rest, we mean it's not firing an electrical impulse. Now, my next whiteboard, I'm going to quickly do this again for illustrative purposes. We know what resting membrane potential is now, what sets it up and what it means. If I were another nerve cell and I were to stimulate the cell that I'm drawing now, the one that's at rest. So let's just say this is me, and here are my axon terminals. And let's just say I release some neurotransmitter onto the dendrites of this cell. Initially, what's going to happen at the receiving cell, it's called the postsynaptic cell because it just means the cell downstream of this junction. So at the postsynaptic cell, we first get something called a graded potential. A graded potential is a short-lived, localized depolarization of the membrane that's dependent on stimulus strength. So I'll say that again. I'm just turning off this room heater in my room. Our furnace went out last night, and we're expecting not that you care, we're expecting a furnace guy to come today. So graded potential, when I stimulate this resting cell, the resting cell or postsynaptic cell exhibits a graded potential. That's a short-lived, localized depolarization that is dependent on stimulus strength. So let's first define depolarization. Whenever you see this word, what it literally means is if on one side we have positive, the other side we have negative, these two polar sides, they're polar now, positive, negative, they're polar opposites. That makes sense. If we create a situation where this side becomes less positive and this side becomes less negative, AKA these two things migrate toward each other like this in terms of charge, that is the act of depolarizing the membrane. So what I tell students in analogy is, there's the Earth, the globe. This is the North Pole. This is the South Pole. Here's the equator. If you were standing here and I was standing here, we would be at polar opposite ends of the globe. If both of us were to travel or migrate toward the equator, we are depolarizing our positions. All right, we're getting closer together. That's what this means. That usually, or I say usually, always equates to excitability. So when we depolarize a cell membrane, it causes something to happen. All right. All right. So now I'm going to stimulate this cell. This is me. And I'm going to release my neurotransmitter onto this resting cell. When I do that, this situation of resting membrane potential, it flips. Localized means right at the area of stimulation. So right at this area of stimulation, I get ripples of depolarization, like ripples in a pond when you throw a rock into a pond, that emanate away from this area. Remember, depolarization means this positive gets closer to the negative and this negative gets closer. Think of two magnets coming together. When you pull two magnets far away, nothing happens. They're polar opposites. When you slowly bring those two magnets, if they're positive, negative sides, closer together, you start to feel energy. And pretty soon, excitability, and something happens, the two magnets smack together. That is the act of depolarizing. When I stimulate this cell, it exhibits something called a graded potential, and there are ripples of depolarization that emanate from this point of stimulus. But if this is a weak stimulus I'm giving, those waves of depolarization die out fairly quickly. If I stimulate this cell with a stronger stimulus, more neurotransmitter, those waves of depolarization are bigger and they travel further, like this. These are graded potentials. Okay. Now, if I stimulate this resting cell, this postsynaptic cell, strongly enough to reach this resting cell's threshold, write that term. Threshold is the stimulation point where this resting cell will fire its own electrical impulse, aka we turn it on, we wake it up. So we're poking this cell stronger and stronger, and it can feel the pokes more and more. Finally, if we turn this cell on or wake it up, here's what happens. Right here at the axon hillock that we defined in our last conversation, it's called the trigger area of the axon hillock. Sodium channels, sodium gates, those protein gates that we defined on the last whiteboard, they open up. So a really large sodium gate opens up right here, and sodium ions literally gush into the cell. All right. When that happens, we've turned this cell on. The sodium gates downstream of this open in the same manner, one by one, all the way down the cell membrane like this. It's called linear propagation. Just think of it as you tip the first domino right here, and then it tips the next one and the next one and the next one. So these sodium channels or these sodium gates open up really, really wide. Sodium ions literally rush into the cell very quickly. That is a massive act of depolarizing this membrane. That literally is what an action potential is. So when you see this term or you hear me say it or you read it, action potential is a massive depolarizing event. And this is what we call the electrical stimulus of the cell. I don't have time really to give you a full lecture about electricity, but when we have a regular battery that you plug devices into, I'm just going to draw an old-fashioned one here. There's a positive end and there's a negative end. We've separated charges within this cell. You know there's a positive end to a battery and a negative end. We've separated the charges. It's polar. When we hook a wire up to this end and connect it to this end, we are depolarizing this situation, and there's a flow of electrons within this wire that were once separated in the battery. That's what gives electricity. It's not that different here. When these sodium gates open at the axon hillock, and sodium gushes in, we get a flow of electrons, a flow of ions across this membrane in a linear fashion. That is depolarization. That is the firing of an action potential. This is an all or none response. What that means is once we open this sodium gate at the axon hillock, we can't turn it off. It happens, and this thing is going to fire all the way down the axon, and we can't stop it. So it's all or none. Once it gets clicked on, you can't shut it off. That's, believe it or not, that's only step 2. Step 3 in this process, I need to do some erasing here to clean up my neuron. So I'm just going to erase some of this. There's my axon membrane again. Let me erase this arrow. So we've depolarized, we've caused an action potential. This nerve cell is woken up, it's reached threshold, and it's firing its action potential all the way down the axon. By the way, when that depolarization gets all the way down here, you know what happens. This terminal end releases its neurotransmitter into the synaptic cleft. All right, after depolarization occurs, the next step is repolarization. So our original situation was sodium outside the cell, potassium inside the cell. We gave it a stimulus. It caused a graded potential to reach this cell's threshold. Sodium gates opened up at the axon hillock. We fired an action potential. Then, as these sodium gates open during depolarization, the ones upstream begin to close. So as these are opening down here, these begin to close up here. Sodium gates, sodium channels. Then the act of repolarization starts, and that's the opposite. Potassium ions that are in here inside of the cell, they start to flow out. So during repolarization, potassium channels open up. That's supposed to be 2 ends. Potassium channels open, potassium flows out. That is the act of repolarizing the cell. The reason we call it repolarizing is because such a huge quantity of potassium flows out that we reestablish the original polarity of a positive atmosphere out here and negative in here. That's why we call it repolarizing the cell. Now we're almost finished with this story of how a nerve cell fires an action potential. So we've repolarized this cell, positive out, negative in, but now we've got a reverse ion situation. We've got potassium ions out here in high concentration, sodium ions in here in high concentration. So it's the sodium potassium pump that then reestablishes, that's supposed to be an S, let me correct that. That reestablishes the original ion concentration. So those little pumps on the surface of our membranes that constantly use ATP, they're called ATPase pumps, they pump sodium out and potassium in, and that's what reestablishes the original ion concentration. We're back to the beginning of the story. So I don't have time to rehash that whole situation again. Watch this video more than once and read your textbook, and it will sink in, I promise. Now, during the act of depolarization, remember that's sodium channels opening up, going like this. During this process, this cell that's firing its action potential is under what we call absolute refractory period. During depolarization, the firing of the action potential, sodium gates opening up, we can stimulate that cell again and it won't respond to anything. So all we're saying is, if this cell is undergoing its massive depolarizing sodium rushing in, and we stimulate this cell again, it won't do anything, it won't respond. That is absolute refractory period. There is something called relative refractory period. There's supposed to be an R there. It's not refractory, it's refractory. Relative refractory period refers to, it's almost the same as absolute, but it's slightly different. So let me go back to my picture. When these sodium gates start to close, as these are opening down here, these begin to close. When these start to close and then potassium ions rush out during repolarization, if we stimulate this cell again, and the new stimulus is as strong as the first one or stronger. So if we stimulate it with a stronger stimulus, it will respond and fire another action potential. So I'm going to go back here. This one occurs during depolarization, and the cell will not respond to any stimulus, no matter the strength. Relative is during repolarization, and the cell will respond to a second stimulus if it's at least as strong as the first, if not greater than the first, usually greater than the first. Okay, that's a lot of heady stuff, and I'm not finished yet, but that's the most complicated issue of what we're discussing today. All right. Now I'm going to talk about conduction. This is a new topic. It's related, but it's a new topic. Conduction velocity. How fast? Conduction velocity refers to how fast does this neuron fire this action potential. All right. Conduction velocity is dependent on two things. First, axon diameter, and second, degree of myelination. Remember, myelin is that lipid-based insulation from Schwann cells in the peripheral nervous system and oligodendrocytes in the central nervous system, that insulation that wraps around the axon. So the larger the diameter of the axon, the faster the conduction velocity speed will be. Also, the more myelinated the axon, the faster the conduction velocity will be. And this is how that works. I'm not going to do all the detail of this, don't worry. I'm going to myelinate my neuron now, which I didn't do in my first drawing. When we have an unmyelinated neuron that's relatively thin, when an action potential starts here, and it has to go from gate to gate, domino to domino. It can't skip any dominoes, okay? And that is like walking across the room, heel toe with your feet. You have to touch the heel of your front foot with the toe of your rear foot in order to move. That's somewhat cumbersome and not very efficient. That is called continuous conduction, and it occurs in unmyelinated neurons. And it's relatively slow, right? Think domino has to hit that domino, 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 domino. That's quite slow. Now, if I myelinate this neuron, and I'll use a different color to show you there. Here's my myelin insulation. And this myelin is arranged so that there are these spaces or gaps between this myelin sheath. We call those gaps nodes of Ranvier, just after the French guy who discovered them. So this open space is called a node. Okay. When we have this myelin on here, it not only helps insulate this cell, but it really speeds up transmission. And here's how it works. Sodium gate opens here during initial depolarization when that threshold is reached. Now this cell doesn't have to go heel toe, heel toe, heel toe like this. The impulse, the depolarizing, actually can take giant steps and jump from node to node. That increases speed dramatically. And that is called saltatory conduction. Saltatory conduction occurs in neurons that are myelinated, and the impulse, the depolarization, the impulse literally jumps from node to node, skipping those places of myelination. That increases speed dramatically. And we have designated different groups of nerve fibers based on their speed of conduction. Group A fibers are the thickest or largest in diameter, and they also have the most myelin. So I'm just going to write heavily myelinated. So these are the fastest ones. These are typically somatic motor neurons. So I'll put that over here. That means motor neurons, that those are neurons carrying impulses away from the central nervous system. Some sensory neurons as well. So I'll put somatic and sensory neurons of the motor and sensory division. They generate speeds up to about 300 miles per hour. That's pretty fast. If you think going from your head or your neck out to your hand, 300 miles per hour, it gets there in milliseconds. Group B fibers tend to be smaller in diameter and have much less myelin around their axons. So they are lightly myelinated. And they conduct impulses, it's about 10-fold less, so about 30 miles per hour. And finally, group C fibers these are the smallest in diameter, and they lack myelin altogether. They're without that myelin sheath. And the decrease in speed is about 10-fold again, about 2 to 3 miles per hour for these. Now, these two groups tend to be autonomic motor fibers. These are more somatic. Somatic, just think going from the central nervous system to our voluntary skeletal muscle. You know that when you stick your finger on a hot stove, it gets pulled away pretty quickly. That response is really fast. As well as somebody like a professional baseball player at the plate, a batter. He can see, 100+ mile per hour pitch coming at him and respond to it in milliseconds. That's group A fibers at work. Group B fibers are autonomic fibers, which are things like the nervous fibers that service our digestive system, our respiratory system. So they're much slower. Now let's say a few things about synapses. We know that a synapse is just a junction. Sorry about the spelling. When I get close to the top of this board, because it's on a vertical surface, it moves when I try to push against it with this pen, and that's what causes me to really mess up spelling. Synapses are junctions between a neuron and some other cell. It can be another neuron, but it can also be a muscle cell or a gland cell. So the synapse just refers to that junction. There are different types, two main classifications of synapses in the human body. There are electrical synapses, which we have not talked about yet, so this is new. Electrical synapses typically occur in the central nervous system, particularly in the brain. So that's their location. And they have a physical connection via plasma membrane proteins called connexons. And this physical connection allows for the flow of electricity from cell to cell. All right, and so if this is a cell in the brain, I'm not going to draw a fancy nerve cell, and here's another cell in the brain, nerve cells, we have a physical connection of these proteins. These things are called connexons. And literally electrical information or flow can flow right across from cell to cell. And the direction of flow is bidirectional. It can go either direction. So I'm going to draw two arrows like this and say this is bidirectional. That allows for more of a synchronous type of communication between cells, and we need that particularly in our brains. The second type of synapse are called chemical synapses. That's what you know and we've been talking about for the last few weeks. Chemical synapses is there's a release of a chemical called a neurotransmitter. So there is no physical connection between the cells. There's a space. There's always a synaptic cleft. And it's when that chemical is released into that space, it diffuses across that cleft and binds to receptors on the postsynaptic cell that something happens. This is always unidirectional. So the communication always goes from the presynaptic cell to the postsynaptic cell. So I'm just going to write unidirectional flow as opposed to bidirectional and electrical synapses. Now we're going to quickly investigate how, let me call up a new whiteboard, how neurotransmitter is released. So here's our, I'm just going to remind you anatomically, here's our motor neuron. There's this is the axon terminal. These little buttons, these axon terminal buttons, magnify it really large. When that depolarization, that action potential comes down the axon on the membrane like this, when it gets to the end, this synaptic end bulb or button has neurotransmitter chemicals stored in vesicles like this. When this electrical impulse travels down this far, there are calcium channels that open on the membrane. So they would be, those calcium channels would be literally classified as voltage gated channels, because when this voltage changes, they open up and calcium ions out here flow in. Now, we said that when we talked about the neuromuscular junction, literally what happens, those calcium ions, they flow in and those ions bind to little receptors on on the vesicle surface. So I'm going to draw that. Let's just say our calcium ion is this blue dot. So it's the channel opens, it enters the neuron, and the calcium ions bind to these protein surface receptors. When that happens, that causes this vesicle to migrate toward the end of the synaptic terminal, and it binds to actually another protein down here on this surface. These proteins, the red ones, let me label them, are called synaptotagmins for synapse. Okay. When calcium binds to a synaptotagmin, it moves, it changes shape and migrates toward this protein, which is called a snare protein. When the synaptotagmin that's now bound with calcium binds to a snare protein, it causes this vesicle to fuse with the membrane and release its neurotransmitter. It is this whole process of starting here where this calcium channel opens, then it binds to the synaptotagmin, then that vesicle migrates to the end and binds to the snare protein. That's what we call synaptic. That whole process is synaptic delay. It is the most time costly activity in the process of the junction, meaning it's the time delay that's most significant in the release of neurotransmitter and the binding of it on the postsynaptic cell. All right, now I'm going to talk about the postsynaptic cell down here just a little bit because your book does. We're making good time, so this is a good thing. You guys should listen to this lecture a million times before the next test. So the postsynaptic cell just means the cell that receives the neurotransmitter, okay? At the postsynaptic cell, there are two different actions that occur when a neurotransmitter binds to receptors. The first type we know well is called an EPSP, and that stands for excitatory postsynaptic potential. So excitatory is the key word. During an EPSP, when this neurotransmitter that's released to the synaptic cleft, when it binds to receptors at the postsynaptic cell, could be a muscle cell, could be another nerve cell. If that postsynaptic cell, one thing that it can do is the binding of that chemical neurotransmitter can cause this cell's membrane to depolarize. That's an excitatory event. So always equate EPSPs, excitatory postsynaptic potentials, with depolarization of that receiving cell's membrane. I'm just going to say the postsynaptic membrane. Remember, depolarizing is excitatory. When we polarize something, we give it the potential to do work, but the closer these things, remember those magnets, the closer they get together, the more force you feel, the more excitatory something's happening, a magnetic field is created. The further apart we move these, that's inhibitory. And that's the next type of action that can occur at the postsynaptic cell is called an IPSP. And this is an inhibitory postsynaptic potential. So it's all language that's confusing, really. The concepts are not beyond the grasp of anybody in this class. It's learning the new words and the new language that's the most difficult. So back to our example, if we release, let me erase some of this business. If we release a neurotransmitter onto this postsynaptic cell where these receptors are that I'm drawing, and it causes the membrane not to depolarize, but to hyperpolarize, that means if we have positive, negative here, rather than those two charges getting closer together, they get further apart. It hyperpolarizes the membrane. Just like when we were standing on the globe. If you're here, North Pole, and I'm down here, South Pole, we are at polar opposites. An excitatory response would be, let's say you and I like each other and we get closer together. That's an exciting event. That's depolarization. Hyperpolarizing would be if we get further apart. That's hyperpolarizing. I ran out of space. And that is inhibitory. So when this neurotransmitter binds to these receptors, if it hyperpolarizes this membrane, that means we're actually inhibiting this postsynaptic cell from doing something. We're making it more difficult for it to do something. Now, what causes a neurotransmitter to either be excitatory or inhibitory is not the type of neurotransmitter itself, but the type of receptor at the postsynaptic cell. So whether or not the event is excitatory or inhibitory does not depend on the individual neurotransmitter. It depends on the type of receptor at the postsynaptic cell. Classic example I can give you of that is this. We know that acetylcholine, so example, acetylcholine causes skeletal muscle cells to contract. We know that. So it causes a depolarization and it's excitatory on skeletal muscles. When acetylcholine is released on cardiac muscle, guess what? It's inhibitory. So acetylcholine on our skeletal muscles it's excitatory and it causes them to contract. Acetylcholine, the same neurotransmitter, when it binds to cardiac muscle cells, it causes the heart to slow down and it inhibits contraction. It hyperpolarizes the membranes of those cardiac muscle cells. That tells us that excitatory versus inhibitory is not dependent on this, but on the type of receptor at the postsynaptic cell. We can summate the response. I'm writing temporal summation versus spatial summation, and we already went over these examples when we talked about muscular contractions. Temporal always means time. Summation means something that's additive. Spatial, think of this as the number. Summation means additive. So when we are stimulating a nerve cell, there's a nerve cell at rest. If I am a presynaptic cell and I release my neurotransmitter onto its dendrites, the cell body is also, the soma is also responsive to this, receptive to this as well. If I reach this cell's threshold, it causes an action potential to fire. We just went over all of that. If I stimulate, take a break, stimulate, take a break, stimulate, take a break, this will fire, it will relax, it will fire, it will relax. If I increase my rate of stimulation, meaning I stimulate that cell faster and faster, stimulate, 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 stimulate a lot faster, notwithstanding the absolute refractory period of this cell will fire an action potential faster, faster, faster. Obviously, if we reach its refractory period, it will have to break for that. That is the act of temporal summation. The rate of stimulation increases. That increases the firing rate of the neuron. Now, spatial summation means the number of neurons that are actually stimulating this cell. So let's just say, I'm going to get my eraser out here and get rid of this business. Get rid of my temporal summation. I'm stimulating this cell, and let's say somebody else in the class is a neuron and they're stimulating this cell, and somebody else in the class is stimulating this cell as well. And all three of us are stimulating this cell at the same time. That is spatial summation. That will also increase the firing rate of this receiving neuron. And that refers to spatial summation. By the way, here's a mind blow. This is very simple down here. Some neurons can have up to 10,000 synapses in one neuron alone. So that gives you a brief glimpse as to how crazy complicated our nervous system really is. Billions of neurons, and some neurons can have up to 10,000 different synapses. The numbers are crazy, they're mind-boggling. All right, so we're almost done today, which is getting to be the good news. We've talked about both excitatory and inhibitory, spatial and temporal summation. Now I'm going to talk about neurotransmitter receptor types, those receptors on the postsynaptic cell. I'm just going to list them and briefly talk about them. There are channel, that's supposed to be two N's. I'm at the top of the board again, so it's cumbersome. Channel-linked receptors. I'll put a number 1. I'll list these as numbers. These are types of neurotransmitter receptors. These tend to be ligand-gated ion channels. That means chemically gated. So a ligand is another name for a signaling chemical. And when a chemical binds to that receptor, it causes something to happen. These protein receptors, they change shape when that chemical binds to them. This is very typical of hormone type of action in the human body. These type always receive neurotransmitter. They can be either excitatory or inhibitory. The key is that they are activated by a chemical binding to the receptor. And you know now they can be excitatory or inhibitory because I gave you examples of skeletal muscle and cardiac muscle cells with the neurotransmitter acetylcholine. They have two different types of receptors on those cells. Both are channel-linked receptors. There are also what we call G protein linked receptors. Since you haven't studied the endocrine system yet, the endocrine system uses this as well. I don't expect you to really have a solid grasp of what these are. These are more indirect. They're not quite as direct as channel-linked receptors. These receptors are much slow. I'm going to write the word indirect. They're also much slower acting than channel-linked. The reason is because there's more events that have to occur. Rather than, in this case, up here, if we have a channel-linked receptor, there it is. If a chemical binds, here's a chemical, if it binds to this receptor, it opens up and things come in. On G protein linked receptors, typically there's a surface receptor protein, but it's linked to another protein inside called a G protein. And that's linked to, it causes this whole chemical response to occur within the cell. There are many more steps. The chemical, in this case, has to bind here, and then it activates this, which activates usually a cyclic AMP as a second messenger. There are several steps involved, and so it's much slower acting. We tend to find these G protein linked receptors, they're also called metabotropic receptors, in central nervous system, particularly in higher learning processes. Just like this course, it may take you a bit of effort and time to learn complicated material, and it's a slower process. Last topic of the day, here we go, and that is injury and repair. I'm going to briefly talk about injury and repair of neurons. First and foremost, there's details in your book about this. I think they have diagrams as well. This is pretty much an absolute. In the central nervous system, the brain and spinal cord, there's basically no repair of neuron injury. Now, I have to put a disclaimer on that. There is, in most recent scientific research, there is some lab in situ evidence of, I think it's mice nasal receptors. So that's in the central nervous system that are exhibiting some evidence of repair. But just for this discussion, just remember, let's just say that there's absolutely no repair in the central nervous system. So once a neuron in the central nervous system is damaged or dies, you're not getting it back. Good thing that we're born with billions of them. I'll tell you why that is in a minute. But in the peripheral nervous system, there is repair. It is largely dependent on the degree of injury. So the amount of repair is dependent on how injured that neuron is. So I'm going to quickly draw, and we're going to injure this neuron. If, for example, there's a complete severing of this axon, and by the way, it's these fiber tracts axons that get injured because they take up most of the space, the linear space in our nervous system. If there's a complete severing and say the broken end of this part of the axon is displaced from the broken end of this one, chances of repair are really minimal in this case. And if and when it does repair, what we often get is sometimes nerve collaterals then grow off into a direction where the original axon did not appear. But however, if this is the axon and it simply gets crushed or depressed and there's still some neural tissue continuity there, there is a large propensity for this nerve, this axon, to be able to regenerate or heal itself. And this is basically what happens. We call, when this nerve is injured, there's a degenerative process that occurs first. And the axon that is distal, some axon tissue distal to the cell body out here dies, and this one would die as well if the cell body is this way. That is called Wallerian degeneration. Degeneration. So basically, phagocytes come into this injured area and start cleaning up debris. Neural tissue dies. Macrophages, phagocytic macrophages come in and start digesting that dead tissue. Schwann cells, however, that make up the myelin sheath, let's color code this. Schwann cells do not degenerate. Especially during times of injury, they actually proliferate. They have this ability to respond to injury by proliferating, which is a good thing. They're the key factor really in neural injury repair. And they form a bridge like I've drawn here and here to help start the repair process. So those Schwann cells proliferate and they form a bridge with the original part of the axon. And then the Schwann cells start to secrete something called NGF, or nerve growth factor. It's a chemical that stimulates nerve fibroblasts to become active. That is really the key to nerve repair. Those nerve fibroblasts can also secrete NGF as well. So these also secrete more of that nerve growth factor. Now back to our story about the central nervous system. Regeneration in the central nervous system is almost never, super rare if ever at all. And it's not only due to the complicated environment of the central nervous system, but remember, it's oligodendrocytes in the CNS that form myelin, not Schwann cells. So oligodendrocytes, they're what create the myelin sheath in the central nervous system, and they do not have this regenerative capability. They do not secrete nerve growth factor, and it's therefore theorized that it's because these cells exist in the central nervous system myelin, and these cells make up the peripheral nervous system myelin. That's the big difference in why central nervous system tissue regeneration is almost non-existent. All right, guys, I'm going to cut it there for the day. So let me cut my recording and then I'll take questions.